Hi, everyone. I'm Clef from the University of Otago in the South Island of New Zealand. So I've come from winter to summer. The gauntlet was thrown down by Jennifer. She wants perspectives. So I thought I would I'm a guinea pig, obviously, so we'll see how this goes. I'm just going to show a couple slides. I'm going to ask a question. I'm going to give an answer for one set of synapses, one part of the brain, and maybe I'll make some discussion. So here's the question. LTP, input specific, LTD, typically input specific. Synaptic tag and capture just brings more synapses in, but not all the synapses. So how does network stability be maintained? How is it maintained if you have input-specific change? And if you can just have LTP, and that's often shown, I think Tom showed a very good example of a second pathway that hadn't have any LTD. If that happens in real life, how do the networks survive well without becoming too hyperexcitable or too hypoexcitable? And the classic answer that would have been sort of perhaps touched on by Matthew this morning is that the kind of homeostatic plasticity answer. So if you have too many synapses strengthening, then you have homeostatic downscaling. Everybody goes down, so the net input weights are back to the same, but the differences between synapses are maintained. So that story has been around for quite some time with basically no evidence, which is not to say it can't happen, but basically no evidence. So what I want to show you is some old data. First, doing an in vivo awake animal LTP experiment in the dentate gyrus, stimulating the medial and lateral perforant path inputs to the dentate gyrus. And actually, this is work I began when I first went as a postdoc to New Zealand. So carried on, and basically, so you can the 2-pathway experiment, like we just heard about. You can alternate stimulation between the 2 pathways, and you can produce LTP. Let's say on the medial perforant path, fine, and without doing anything evident, LTD occurs on the lateral perforant path. And you can see that we gave 3 different protocols, 2 LTP inducing for the medial path, and 1 for the LTP on the lateral path. And basically, if one goes up, the other goes down, even though that we're not intentionally stimulating the heterosynaptic pathway. So this was traditionally been called heterosynaptic LTD, all right, and activity-independent plasticity. And this is just showing the recordings. What you can do, of course, if you're recording in vivo of implanted electrodes, you can record over time. So this is over a 2 weeks period with these different protocols, and you can see that they more or less decay at the same rates as well. So the brain has figured out, in this system anyway, an online balancing of LTP and LTD in these networks. Now, what I'm not going to show you is that actually, this is actually an activity-dependent LTD, because if you silence that pathway while you're giving this high-frequency stimulation, we do delta burst stimulation here in these experiments, then you don't get the LTD, but you get the LTP. So the constitutive activity in the system is driving this LTD.
So Cliff,
go.
So this is plasticity for long-range connections, right?
Yes, so well, that's right. We're stimulating in the angular bundle. It's a long axonal projection from entorhinal cortex into the dentate gyrus.
So I generally think of network stability having to do with local recurrent connectivity. So this is just, the input strength, right? That's not going to tell you whether the network itself is stable, whether it's epileptic.
Well, it may become, this is only one synaptic pathway. This is happening throughout the circuit. Who knows in real life? Of course, we're just parceling out one synapse. And of course, if the input has become unstable, unusually activatable because it's too strong from before, then that might drive network instability. I'm just projecting here. Okay, I haven't done modeling to kind of show
I
that.
guess it depends. If it's a general phenomena, it occurs with local recurrent connections, that would be useful.
Yeah, but I mean, just off the top of my head, I don't think you would have to only think about recurrent connections to think about stability of the network. If you, if the inputs, I mean, these inputs are, you know, they're active at more or less at different times during behavior, and so if the input is too strong and the cells are firing too much, and who knows what's happening to inhibition at the same time, yeah, who knows?
It might be better to think about it as saturation of stability. You still have the problem, you don't want to saturate the system, so all the synapses get too strong. But it won't generate sort of an epileptic seizure.
I mean, the stability has to do with loops, and eventually it loops back, right? But
Yeah,
it can take a long time.
so there is a circuit going back to the entorhinal cortex. Yeah, I take your point, and in some ways, yeah, keeping away from saturation was another objective. Okay, so you might say, okay, that's all very, very well. Yeah.
So there's other people have shown data that the hyposynaptic LTD is dependent on ongoing activity in the hyposynaptic pathway.
And we've shown that here too.
And so in this case, if you stop stimulating the other pathway, S2 pathway, let's call it, it won't undergo a.
It's not the stimulation that's driving it. It's some endogenous activity. So we actually put procaine into the lateral path, prevent it from even having endogenous firing, and that blocks the LTD. So, then there's, you know, we've done some computational modeling to kind of explain this through metaplasticity mechanisms, but that's not really what I was going to talk about, although happy to if you want to. So then, many years later, I developed a collaboration with Kristen Harris, who has been mentioned before, and she wanted to look at this tissue, not the previous animals, of course, but new tissue, where we induced LTP in the medial perforant path on one side of the brain and control stimulation on the other side of the brain. As you know, probably Kristen does 3D serial section EM reconstructions of dendrites and spines and now astrocytes and, you know, everything that they can see. And then look at the distribution of the spines. Now, Matthew has also mentioned the log normal distribution of spine sizes. These are the spine head volumes. And you can see that, you know, this is also true in the dentate gyrus and the termination zone of the medial perforant path. And it expands out after LTP induction, and you get this kind of distribution where you have more of the larger spine heads. But interestingly, you can see that there's actually more of the smaller ones as well. And when you plot, you do some kind of, you know, analytics on this, there's no change in the number of spines, so you can't really see that very well. I'm not even sure what that one is. But the main thing is if you look at the PSD contact area per length of dendrite, right? Then there's a balancing of the increase in some spine sizes and a decrease of others. So there's no net change in the PSD contact area onto that dendrite. And that's true at both 30 minutes, so it happens very quickly, and 2 hours post LTP induction and LTD induction. Oops, so there we go. And that's my point. So there's different plasticities, as we've been hearing. There's the Hebbian plasticity, fast, acting, enduring, input-specific, cooperative changes, the synaptic tag and capture story. And we've also been hearing about homeostatic plasticity, slow, maybe non-enduring. Non-specific is probably wrong now, so I should probably take out because some spines are changing and not others. And there's, you know, other types of homeostatic plasticity as well. But there's this other types of synaptic plasticity that serves, it seems, a homeostatic function at the non-tetanized inputs. And as I mentioned, without showing the data, it's dependent on the rate of activity. And that's my story.
Thanks.
Yes.
So just on the previous slide, I wasn't carefully following. So it's the area, it's not the volume that's actually the sum.
This one is about the PSD area.
Yes.
Right. So it's they've drawn around all the PSDs three-dimensionally. They've got the sizes, and they calculate the micron squared per micron of dendritic length.
Uh-huh.
So I don't think we've done it with
the volume. If you would do the volume, you might see different.
You might see different with volume, although I think there'd probably be a balancing there as well. But the main thing is really not how big the spine heads are, it's how big the contact areas are, which is related to the number of AMPA receptors, which is related to their synaptic strength. So, yeah.
Go ahead.
Yeah, thanks for that. The thing I like about heterosynaptic plasticity is that I think maybe the more important problem isn't the spatial scale and the input sensitivity, it's the time scale. And that maybe the homeostatic changes might just be too darn slow to compensate for the change in excitability induced by LTP. And so kind of co-expressed, or very shortly thereafter co-expressed LTD might do a good job kind of compensating for stability or saturation, or however you want to think about it.
You're talking my language.
Yeah.
That's exactly how we're thinking about it. I mean, to be honest, okay, so I'm darts and daggers coming. Homeostatic plasticity, in my view, is response to pathology, right? 2 days of TTX, right? Eye binocular deprivation or whatever over prolonged periods. So this is part of the nervous system's response to prolonged abnormal activity levels, whether it's up or down. And that's part of its toolkit, to be fair. But I think it needs, hopefully Tara Keck's going to support me on this, but I think it needs a faster acting balancing process as well. And to remove the need for the previously described homeostatic plasticity, except for extreme circumstances.
Yeah, I mean, I think that's the issue, right? It's like, how little activity do you need? How brief a period of deprivation do you need? Only in a typological prolonged deprivation, or a few seconds do it. How does that know?
Yeah, I think Gina's done like an hour-by-hour analysis, and it happens within a couple hours.
That's still slow.
But that's still pretty slow, and yeah. So the main thing so when we did computational modeling of this, the important thing for the model was to have spontaneous activity in the input lines. Otherwise, it didn't happen. Just like if we blocked the activity with Procaine. So a lot of models, computational models of plasticity, in the past anyway, haven't had spontaneous activity. They have a learning rule, there's activity that drives the learning rule, but then it's like there's nothing before and after. It's just what happens there and what the change is and modeling that. So, and I think that's moving away. I mean, I don't really follow that literature hugely, but I think having ongoing activity at these synapses is quite important for understanding how the cells responded.
Yeah, go ahead.
I have a question. Thanks for sharing this data and beautiful in vivo recordings. I mean, these are the nice time axis over days, right? But what you showed is that actually the difference you induce by the some pathway potentiated getting depressed is really like a contraction back to the original situation really over weeks, right?
That's right.
So what also beautiful talk earlier today, but these were these heterosynaptic homeostatic changes are still, if I got it correctly, within hours or much shorter time scale. So my question is basically, what's your hinge on these late processes happening over the days where you observe this washout of this initially imprinted differences in the synaptic weights that are gradually contracting back to the original situation? What's the mechanism there?
Well, frankly, we don't know whether it's an active or passive decay. We have seen examples where it lasts for months and months and months, so in the same system. So it can last for a long time. And we have shown in previous studies that if you put the animals in an enriched environment, the LTP comes back and the LTD comes back as well. So, so there's activity drivers that can cause active decay. But in these experiments where we're not deliberately doing it, except giving test pulses, and maybe that's enough, I don't know, that, you know, whether it's, just that we haven't caused enough gene expression, protein synthesis to really last a long time, or whether there's some active decay process. So, yeah, sorry. It's hard to give drugs and things over weeks to sort this out.
How is the set point determined?
How's the set point determined? There have been experiments where the set point's been changed. I think cortical studies. I think John Donahue's lab has done this. So, for example, they've done experiments some time ago now, but where they produced LTP in a cortical pathway, saturated it, couldn't get any more LTP, waited some time, and they could get more LTP as if there's some kind of, you know, change in the set point. There's other ways to interpret that from that data. Of course, you know, these animals live in a very boring environment in their normal standard cages. Whether that is setting a set point differently from animals running around the wild, you know, couldn't say, but yeah, we don't really know if there is a set point or whether it's a modifiable set point, but there's some evidence to suggest it might be modifiable. I think I should hand back to the panel at this point.
Actually, I would like to have your thoughts about the possibility that astrocytes could also be involved in this balance and heterosynaptic plasticity, because several studies have shown that they could mediate, and more on the presynaptic side, actually. So you elicit LTP at CA3, CA1 synapses, and you get LTD at non-stimulated synapses, but with a presynaptic LTD. So did you look at that in this set of experiments?
Not in these experiments, but I think, you know, Tom might have something to say. But you have some thoughts on this too, don't you? But astrocytes. But what I know about astrocytes and LTD is more about short-term depression, not long-term depression. However, wait till tomorrow, and I will talk about astrocytes and the control of plasticity. So I won't give it away tomorrow. Thanks, everybody.
Thanks, Cliff. Anybody else have any commentaries? Jennifer? No. Ask away.
For me.
I think maybe more for Matthew.
Oops.
I would love to hear you or anybody in the room speculate more about the function of what you've seen, because I'm just trying to think it through. A synapse undergoes LTP because it learned something, and then that synapse's activity goes down because it's the animal, the brain is doing something else, and then you get the homeostatic increase. So now when you put it back in the context where it's going to express what it learned, that synapse is even stronger. And how do you think about that? I was like making up stories in my head, but you've thought about it for longer.
Yeah, actually it's not an easy question, but so to come back to what Cliff was saying, so again, this is not physiological situation to have, you know, complete blockade of activity for 48 hours. So I think we are mimicking a pathological situation, and I don't know, my vision is that in such a situation, maybe the brain wants some stability. They want to maintain the information stored across synapses. So this could be a way to secure somehow the important synapses, maybe. And my guess, we didn't really look at that, but my guess is that maybe by waiting even longer, maybe we should see a disappearance of some maybe smaller protrusions, or a change in the dynamics of over synapses. So maybe you want to stabilize the very important ones, and maybe that's one. I don't know, that's the way I see things. And then I don't know, in short, I mean, more physiological situation, I don't think you have this you know, pooling plasticities, Hebbian plasticity on one side and homeostatic plasticity at the same synapses. I think maybe it's more the set points of plasticity or that is changed, but in a much more rapid manner, I would say. I don't know if I'm clear or not.
Yeah, and I mean, I guess I was wondering, could this be something related to consolidation of learning, or memory updating is one of the sessions we have, and you know.
So that's another thing, is that, I mean, it would be interesting also to look at this in the context of sleep, for instance. And we know that there is downscaling, for instance, during sleep, which is not uniform actually. So the synapses, I mean, the stable synapses, maybe the synaptopodin positive ones, they are spared by the downscaling that is occurring during sleep. So this would go in the sense of what you are saying, that you consolidate the learning.
Thanks. I've got a question for Tom. So you saw pretty different things depending on whether or not you used a 10-second pulse or a 20-second pulse. And those are kind of just guesses about activity patterns. You think there's a more principled way of kind of choosing your parameter set? So you don't have to just brute force everything and survey the full range? I mean, you mentioned, you kind of alluded to this at the end. I think a lot of us who've worked on this have also been daunted by
so I mean, that's a really good point, and it's something I've thought a little bit about, and see what people think about this approach. One idea, and I have some colleagues at UCLA, Meta, who does in vivo recordings of, you know, firing of CA3 and CA1 pyramidal cells while animals are exploring a novel environment. I'd love to get a big long stretch of that stuff and then run that through stimulating electrodes and use that as sort of a pattern to look at burst-dependent plasticity and synaptic interactions. It would be another way to go. That gets away from just arbitrary, you know, frequency and time. So that would be one approach. And the other thing, you know, the other idea is, you know, I always use 5 hertz. I think that's what physiologists call theta, right? Theta burst, theta pulse, whatever. Theta is 5 hertz. But really, in vivo, theta is a range of frequencies from like 4 to 12, and most common 8 hertz activity. And so that would be another parameter space to explore, right? But another way of getting away from fixed time, fixed frequency, or varying time and fixed frequency, go the other way around. And I played a little bit with that, and you see some, you know, weird things. You can actually like get, you know, using one input to get the cells to enter the complex spike bursting mode, and then do like a frequency downshift. So go from 5 and then shift the frequency, keep it going, go to 3, and the complex spike bursting just kind of explodes. It's amazing how it's facilitated by that frequency dependent shift, right? I don't know what that means. That's like playing around with the slice and seeing what happens, which is an easy trap to get into sometimes. But there's a lot of things, and I think having a more rational, something based on something like in vivo activity, spiking from individual cells, and using that might be a good way to go. Could be informative.
Or starting with a single cell and thinking about interactions between different ion channels.
Yeah, right. Well, I think that's
complex
where
spikes.
so much is coming from that, right? So you're thinking about the activation, especially inactivation kinetics of, you know, voltage-dependent sodium channels, voltage-dependent calcium channels, which are in the dendrites, especially the A-type potassium channels, and how they activate and inactivate, and how they regulate dendritic excitability in response to synaptic inputs. Seems right for sort of computational analysis. Sort of approach would be helpful as well.
Yeah, maybe I don't know about this, but do you see, if you would change the frequency, would you see like in the range of the distance of the connections that are strengthened or weakened, do you see a difference?
I haven't looked at that, but I think that's an interesting question in the context of how the synapses are interacting with one another over distance. So you have apical dendrites, you have proximal apical versus the distal, and then as well as the basal dendrites. How do they talk to one another? And I think they talk to one another through electricity. It's the complex spike burst. And I think Christine has some data on this. It invades everything, right? Once the cell's bigger, curve off a burst. If you look at calcium changes, it changes everywhere, right? And so it's a signal that could go across large spaces. Oh yeah, that would be really interesting. I think based on sort of what's known about the electrophysiology of dendritic excitability, I think the apical and the basal dendrites of pyramidal cells are likely very different. And so frequency dependent activation of different voltage dependent conductance, ion conductances will have very, very different effects. So there's a basis for thinking what you're thinking about is actually potentially really interesting.
So I have kind of a naive question, being a cell biologist at heart, I guess. And that is, there's sort of I've heard two contradictory kind of ideas. One is that a cell is computing how much LTP, positive feedback kind of plasticity it has, and then compensating for it with homeostatic plasticity. So it's all computed in a cell, and you could understand what's happening at the level of cell biology. But then there's this other sort of discussion interleaved here that it's, a network property. So I guess the question, I'm confused about how to think about this. In my little brain, it's easier to think about something that's happening just in a cell. But can you explain all of this stability at the level of what happens in a single cell? Or do you need to invoke changes that you can't explain from things happening in a single cell? When I heard about spines, some shrinking and some expanding, that sounds like a single cell making a computation. It's got to know that, there's intricate things happening just at the level of that cell, but it seems so much more intricate if it's happening at a network level. So can you explain all of this at the level of single cells, just processes operating in individual cells? Is that that's for anyone who wants to speak to it on some level. So I think what you're thinking about is really interesting. I think about it that way as well. And I think we have to, just as when we think of plasticity, short-term potentiation, long-term potentiation, LLTP, late LTP, like really long-lasting, and there's different mechanisms that are producing potentiation over different timescales. I think for homeostatic plasticity, maybe it's the same thing. That some of the things we saw earlier, they might take many hours or a day or two to develop, but that's one temporal component of homeostatic plasticity. And then what Cliff just showed us could be a mechanism, a very rapid mechanism of homeostatic plasticity. And that both at a systems and a cellular level, the circuit's got it covered, right? We can control plasticity using homeostatic plasticity through multiple, either cell biological or circuit-based homeostatic plasticity mechanisms that also operate over different timescales. And I think the rapidly induced homeostatic plasticity, I think, are the interesting ones because we know so little about that, right? And who was it? Wolfram Buscher? I'm out of my league here. The computational person. There was a lot of talk about this amongst the computational people years ago about what they call it, the homeostatic Hebbian temporal paradox. And that was, we've got all this homeostatic mechanisms, but they're slow, take hours. And if you model Hebbian LTP, it gets out of control very quickly, both saturating potentiation or saturating depression, arguing that there had to be additional mechanisms fast that could control it. Are the faster processes cell autonomous and the slower ones more network involved, or is it the opposite way around?
Yeah, I don't know. I think that's a great question.
Actually, I agree with you that the interesting part is the rapid mechanism for homeostatic plasticity, and my vision is that maybe this is occurring by balancing different synapses, and I don't see one synapse, with LTP and homeostatic plasticity coexisting. So there is really a problem of time scale. This is for sure. Then in terms of cell autonomous mechanism, I think this I'm not so sure about this. And I think I wanted to launch a little bit the discussion on that, but I think we should also consider the potential role of astrocytes as a way maybe to regulate homeostasis on different spatial scales, and also potentially at rapid scales, because they react to activity, and the single synapse level, they can also affect multiple synapses across the network, and also at the level of a single neuron. So I think the cell autonomous mechanism could be very important when you modify the activity in the long term, and because the cell has to probably to maintain a firing rate or the synaptic transmission at the level of individual synapses. But I think at the network level, you might have more complex mechanism, potentially involving astrocytes also, or the inhibitory networks as well.
Yeah, Peter. Yeah, so something else that Wolfram was very interested in, indeed, is inhibitory plasticity. So I think all you've been talking about so far has been plasticity in the excitatory system, right? And so, how do you think about the inhibition system? Do you think it's different between the hippocampus versus the cortex? And how do we think about these homeostatic mechanisms in that case?
I think it's much less known what we know about inhibitory synapses. I think there are some studies showing homeostatic plasticity at inhibitory synapses, so they can adapt also, but we don't know about the interactions much. I mean, I don't have in mind, a study looking at both at the same time in the same paradigm. So usually you play with overall activity by blocking inhibitory transmission or excitatory transmission, and then you look at either inhibitory neurons or inhibitory synapses and excitatory synapses. I think this is a key question, and this should require really investigating both at the same time to see the interplay. So I cannot say much more about that because I think we are missing data for this.
Would it be different? Could it be different between the hippocampus and the cortex?
I mean, it could be. Probably there is a balance. The excitation to inhibition balance could be changed to maintain the firing rate. This is for sure. But again, I don't think this has been really. People didn't really look at that in the same state of that. I mean, when looking at synapses, they didn't look at both inhibitory and excitatory synapses because I guess it's not easy to.
Thank you.
Sorry, so we've done that experiment where we look at E and I on the same cells in vivo, and the inhibitory changes really rapidly. The excitation changes much more slowly. We see inhibition. You can see changes if you think about it at a network level, which is how we think about it in vivo, where cells are embedded in networks, and you don't have a complete removal of activity. So no matter what you do, you can remove all feed-forward activity and in mouse cortex, you get to about 60% of activity with feed-forward. So you have a reduction of activity, but not a full removal as you would get with TTX in a culture. So that's, if you took away all activity, the mouse would die, I assume? I don't know. But I think that, yes, so you have a rapid reduction of inhibition, and then the response of the excitatory synapses that are strengthened after that, in that EI balance has shifted, and that slowly goes back over time, and that's on the time course of where we see excitatory activity rebounding, and the inhibitory cells and the excitatory cells rebound as a group. So if you think about it in a network way, you have very, very rapid ways to prevent too much activity, because from a homeostatic point of view, too much activity in a cortex is way more dangerous than too little. So too little activity is not going to give you into a pathological seizure-like state. So the inhibitory mechanisms to shut down activity are fast, and you can basically do that through network connectivity that already exists. Whether that's homeostatic or not, I think that that's open for debate. But the mechanisms that kick in at a subcellular level are very much inhibition than excitation in adulthood. What we're talking about probably here is much more developmental. When we think about cultures, it's not exactly clear how to map that, and what we see in juvenile versus adulthood varies quite a bit, but you still are seeing the sort of a reduction of inhibition followed by changes in excitation, but inhibition is weaker in critical period in general. It is already inhibitory, and so there's less of that downturn. So in general, inhibition is playing a huge role in both juvenile and adulthood, and does get at some of these very fast timescales to prevent pathological detrimental activity for the system in cortex. Those experiments have not been done in hippocampus.
A question? Okay. Sure.
Yes, I have kind of a question just looking for some wild speculation. So some of us have been here discussing statistical learning in the brain for about 4 weeks now. And so at the behavioral level, a lot of it, is looking at you experience a series of images and sounds, and some of them are reliably related to each other, and that people are pretty sensitive at picking these up. And what I'm kind of curious about is, like, how do you think about this eligibility trace in terms of what are the types of things that it supports learning and what not? And so I know it's really hard to kind of go from the synaptic level to behavioral level, but I'd love to know your thoughts.
Can I add to that question?
Yeah.
I got it, Mike. Usually you need an error signal. And does anybody, I mean, besides dopamine, does anybody think about error signals?
So
it's just related, presumably heavily related to that question.
We'll hear more about this, I think, tomorrow. But the idea is that, activity ongoing, it's feed forward synapses onto a pyramidal cell can set an eligibility trace. And then you have feedback information that can be related to basically an error signal, instructive signal, that comes into a different dendritic compartment, into the apical tufts of cells, and that triggers the plateau potential within the dendrites, the postsynaptic somatic complex spike bursting. And so it's a feedback error signal, instructive signal that's then gating, right, plasticity induction based on whether there's still an eligibility trace present. So those ideas, I find it really remarkable, this idea of an eligibility trace. It's over 50 years old. There's a paper published in Nature in 1968 by T.J. Crow, it's a 1.5-page paper. It's all speculation. There's not a single piece of data in it. But he proposes this idea of activity within circuits, setting up a silent trace that then gets updated in response to reward, right? So things like norepinephrine or dopamine providing the signal that then changes synaptic strength, sort of implements a Hebbian type of plasticity. So the idea is really old, but it's been around, at least amongst the computational people, I think. Now I'm out of my league, Peter.
Well, yeah, I mean, we always have an error signal whenever we do anything, but we don't know where it comes from.
Yeah, well, what about reinforcement learning?
Oh, for reinforcement learning, yes. For reinforcement learning, yes. For
there you go.
Other kinds of learning, no.
Time from circuits are active, synapses are doing their thing, and then the reward comes later. Well, then that's the signal that.
Older than 60. Older than 60. So how old from a psychological point of view?
Peter?
Yeah.
Microphone. Oh, it's okay. Dean?
Sorry, just want to go back to both Richard's and Peter's questions. So I think it has to be both, Richard. So I think there has to be cell autonomous mechanisms and clearly network mechanisms, whether those network mechanisms are balanced specific inhibition or through astroglia. But I also think it's pretty clear at this point that there is differences between hippocampus and neocortex. I mean, at least even in the excitatory synapses, the learning rules seem to be quite different. For example, in terms of STDP at E to E connections, I'll give an example of that tomorrow. But regarding the stability of how to do the EI balance, there's some data, and we have a bit of this recently, purely on a computational level. It's very interesting. In one way, what's more mathematically stable is if you do homeostatic plasticity to a set point to the other population. So it turns out mathematically that if you want to stabilize activity in a loop, in a recurrent network, to adjust the weights onto your excitatory synapse, you should use the error in the inhibitory neurons. And this relates to this paradoxical effect. So, but biologically, that can happen. It's non-local, but still biologically plausible. But yeah, there has to be a lot of stuff we're missing on that excitatory inhibitory loop and balancing. And I totally agree with both of what you and Rob said in terms of we have to get out of this framework of thinking that there's one mechanism. There's probably one mechanism at the 10-second scale, another mechanism at the minute scale, and another mechanism at the hour.
I'll just jump in again. On behalf of one of my colleagues at Otago, linking the idea of inhibition with the eligibility trace in the striatum. So let's talk about another structure. So he's shown recently that so the cortical striatal input, you can stimulate that, and generally, if you don't have the dopamine signal, you get a bit of LTD. But but the activity will also cause a pause in the cholinergic inhibitory interneurons. And during that pause, if dopamine appears, pauses for a few seconds, dopamine appears, then that cortical activity produces LTP. So yeah, so there's a way to kind of describe. So it's not that the synapses per se have an eligibility trace, you might say, that there's activity, that they can produce plasticity, I suppose, and the dopamine will hit, but under conditions of reduced inhibition, that's also driven by the activity. So just thought I'd throw that out there.
Okay, we have a slide coming up.
Yep. Hi. Sorry, I just literally copied this into a slide. My name is Elisa Galliano. I'm a manufacturing physiologist at the moment. I just wanted to contribute this to the discussion about interneurons and timescales. This is a COVID pet project for undergraduate students because they couldn't come in the lab. So we did a systematic review of the literature on sensory deprivation, and we did that not in cortex, because it's very anatomically incorrect, in my opinion, to compare visual cortex with olfactory cortex, say, for the number of sinuses. So we looked at plasticity in the first brain areas where the cranial nerves send their information, okay? So that would be olfactory bulb, lateral geniculate nucleus, and superior colliculus for vision, and dorsal and ventral cochlear nuclei. So this is a systematic review. This is not my data. We just looked. We defined search terms and we went through the literature. This is rodents and this is adults. And what we saw is that depending on the duration of the deprivation, from hours to weeks, so this conversation about is it pathological or is it really more rapidly homeostatic, the type of plasticity that the literature reported, and please be aware, we are all very bad at publishing negative results, so there is publishing bias in here, is very different. So just again, say the same thing the Tara said, interneurons go first across all sensory modalities. Ours, there's a lot of low regulation of interneurons, so they're not scaling up, is sensory deprivation. They're not scaling up, they're scaling down. Maybe the arrow here should have a question mark, but overall it should lead to circuit disinhibition. And the studies that looked at excitatory principal neurons in terms of hours of deprivation found them mostly unchanged. At our level of days of deprivation, we start seeing the literature started reporting plasticity in excitatory neurons, synaptic mostly. This was also a lot not synaptic, out of intrinsic plasticity. And then if you start really deprived for a very long time, like a week or weeks, then you have macroscopic changes, apoptosis, tissue shrinkage, and extensive still synaptic remodeling, both excitatory and inhibitory. And of course, the olfactory bulb is weird. We also have a change in neurogenesis. So my 2 cents on this is the network is not the cell, and it really depends what you're doing. I would very much like to know. Sorry, the last thing that we saw that I wasn't expecting, because I'm an idiot neurophysiologist, so I patch neurons and I always ignore glia. It's the biggest and most conserved form of plasticity across all sensory system is glial plasticity. I'd like to see if this is the same for enrichment, and because that's maybe more relevant to animals exploring new areas. Deprivation, we could argue that it's indeed more pathological or transient.
I think there's a paper coming out in the that shows See, COVID should have lasted longer. We could have included. No.
So I've heard anecdotal reports of people losing their smell for months and then it coming back. And usually it comes back a little weird, but it's very quickly established. Everything was back to normal. Is that correct?
It depends on the people, and it's really because olfaction is really strange because the receptor itself, the olfactory sensory neuron, retain the ability to regenerate throughout life. So they can come back, the stem cells are there, it's fine. The parosmia, which is like smelling coffee and it maybe smells like something else, is because of the targeting of the axons to the correct glomeruli. It gets a bit haywire, which is an incredibly difficult cell biological problem to target an axon in an already existing circuit, but it seems to go relatively fast. For some people it doesn't. I mean, I've spoken, all of a sudden olfaction is interesting now. So I've spoken with quite a lot of people that went through anosmia, COVID-related anosmia. There is a spectrum of speeds of recovery. Some people never recover actually. And for some people the parosmia is very protracted. And some people actually speed up the process by doing olfactory training, which basically is enrichment or changing the statistics of what you present.
Why do you say it's a network and not a cell? I mean, this is an overall effect, right?
If it were cell-specific, I would not understand why an inhibitor in the neuron would downregulate this primary rate. From a cell autonomous point of view. Like you're receiving less input, molecules are firing less. So many of these mechanisms, and this is the literature, this is what we see in my lab, we really are at this level. We see a lot of intrinsic plasticity of intrinsic excitability, which is also accompanied by axon-short segment structural plasticity. So just have more
but that's one part of the many different kind of things. So.
And even inhibitory neurons, right? You get less input, you fire less. It can be completely local. I don't know if you call it what you call network effect, but
they don't necessarily get less input.
Well, they must have some signal to.
Yes. I think it depends on the interneuron and also the fact that interneurons, also one of the main differences always if you take into account in thinking about this is that most interneurons are pacemaking. So they retain a firing rate at rest that is independent of synaptic input. So I think that they have a bit more scope to regulate also in partially independent. Also, I do not see the same phenotype in all cells. Yes, not all the neurons.
No, because actually, just by the talk I heard this morning, for example, this is actually one question I'm having. So we had this SP positive and SP negative kind of cells. We're talking about timescales. I was thinking like if an SP negative synapse can, for example, transition to an SP positive synapse, this might be an effect which is not on the same time scale as the size of the synapse. For example, a positive SP synapse can change. And so this might be a molecular pathway how actually timescales, different timescales for plasticity can work.
Yeah, I think you're right. I think it's very interesting. I mean, you may have plasticity that you cannot record with electrodes. So you could have a molecular plasticity where synapses, that's what I believe actually, that you have several biochemical states and transition between those states, like that will have an impact on the ability to undergo plasticity, and this is what is difficult to characterize because you don't see the molecules. But this is what I believe. So in the case of synaptopodine, I believe that some spines acquire the ability to express synaptopodine by maybe recruiting the messenger RNA and the microRNA at proximity, and so you have transitions probably, and this is part of the homeostatic machinery, I think. But this is in terms of time scale. Of course, you're playing with time with this. So. And it's cell specific, but I think it really depends of the paradigm once again. So in terms of cell autonomous mechanism, if you play with, if you overactivate a neuron with optogenetics, for instance, you will have a cell autonomous downscaling of synapses. But I don't know if it can be generalized to any kind of homeostatic plasticity. I think it's very complex.
So I also like to add that in mice, olfaction is their leading sense, not vision. Yeah. So maybe some analogy with human is not as direct. So it's interesting that you see most change in olfaction. I wonder. And also in this kind of a reduced adult neurogenesis, you mean mostly in the olfactory bulb, or it's also including epithelium? It's only olfactory. Do you also see the shrinkage of the epithelium?
We did not search for it because we were looking in those areas. I would imagine. I think about the literature and say so. Yes.
Because when you have a cold, is it mostly the epithelium that's destroyed? You have to regenerate these receptor neurons, no?
It depends on how long the cold lasts. I have become an expert on the biology. The standard cold does last 3 to 5 days. So this is probably not quite long enough to engage
into your weeks region. Yeah, okay,
yeah,
maybe.
That side is more mechanical, like mucus blockage and things like that.
A mucus blockage rather than the sensory neurons being replaced. Sensory neurons replaced every 2 months, 3 months anyway, right?
I think it's activity dependent.
Also activity dependent. Thank you.
Any other questions? Great. Thank you for the morning session. And lunch is at 12:30.